Okay, so chapter 3, it's about the cell. So in this chapter, we're going to learn what are the parts of the cells, the function of these parts, and we're going to see as well the mode of transport of molecules within the cells. As well, we're going to see what is resting membrane potential and how ion concentration gradients affect the resting membrane potential. And we're going to see a specific function of the organelles. We're going to see examples of sending signals within the cells and what they do, like what leads to the synthesis of proteins, what is cellular respiration, and we're going to see part of the cell cycles, and we're going to outline the events of mitosis and cytokinesis. So the cell is defined as the basic unit of life, and this unit of life has the different components that helps for having independent function, that it's unique. So all living organisms are made out of cells. And then the cells carry essential functions, which includes metabolism, growth, communication, and reproduction. We have a little bit over 200 different cells, and they have different shapes, they have different functions, and they tend to form groups where they form organs. Now, we have between 30 to 40 trillion cells. And these cells are located in specific locations, and they form, as I said before, tissues. And these tissues of different types, 3 to 4 tissues can form an organ, and organs can physically connect most of the times, and they can form an organ system. So all the work that the cells do will be essential for metabolism, for homeostasis and support life. So here we have examples of cell variation, cell shape variation. So with this cell shape variation, we can perform different functions. So for instance, this is a long cylindrical cell that has banding pattern. So this is an skeletal muscle cell. And this skeletal muscle cell contracts or shortens so that we can perform movement. So that's a function of these skeletal muscle cells. In areas of the body, like in our skin, our esophagus, our gut, and our lining of our respiratory system, we have sheets of cells that protect us. These are epithelial cells. In this case, this is a square cell, so this is a cuboidal cell, and it may help to secrete items into the surface of the lining of the stomach, for instance, or the lining of the respiratory system, like mucus, for instance, so that it can lubricate our passageways so that we can transfer food or air. So that's the function of these cuboidal cells. And then we have this elongated cell that has a body, it has extensions that are short and branched, and it has this long extension that it branches at the end. So this is a classical neuron. This is a multipolar neuron. So this neuron receives signals through these extensions that are called dendrites, process that information from these signals that it receives, and then it sends an electrical signal that we call an action potential, and transmit chemicals that has in these endings to other cells so that it can communicate. So this shape is specialized for communication. This is a nerve cell. And then here we have a disc-shaped cell. This is a red blood cell. This red blood cell doesn't have a nucleus. This is the only cell in our body that doesn't have a nucleus. And the function of this cell is to carry oxygen and deliver it into other cells and pick up the waste of these cells, which is CO2. With this disc shape, what it does, it travels to tubules that we call blood vessels. So this shape, it bends as well. That's why it doesn't have the nucleus, so it can be flexible and it can bend. So it passes through these narrow tubules that we call the capillaries, and is helping to transfer really quickly oxygen and pick up CO2 from the tissues. So this cell is specialized for transport. So if we had, for instance, in the blood vessels, like these tiny tubules, the capillaries, like these square-shaped cells or these nerve cells or these muscle cells, the capillaries will become clogged. So a structure function, that's the principle of physiology and anatomy, a structure function. Shape is correlated to the function. So basic processes common to all cells. Metabolism. And metabolism is subdivided into anabolism and catabolism. So basically in catabolism, you break down large molecules into smaller molecules, and in anabolism, you bring smaller molecules or monomers to create a long or a large molecule. And some of these chemical reactions that take place in the cells are what we call oxidation reduction, in which the cells donate electrons and accept electrons. They are coupled, these reactions. So they have to be a donor of molecules, or electrons in this case, an acceptor of electrons. So these are called ox-redox reactions or redox reactions. And these are necessary to generate energy or to synthesize molecules. Also, we have transport. That's another essential function of the cell, to transport. So the cells, they live in an environment, and oftentimes they endocytose, or they bring in molecules so that they can transport them to other places, or they can metabolize them internally. So transport of substances is the movement of molecules into and out of the cells. And then communication. The cells send and receive signals that they use to coordinate their activities. And cells reproduce. Most of our cells undergo mitosis, and they make other cells. And they use this to either create new tissues, to grow, or to repair. That's the function of mitosis. So, what are the different parts of the cells? Will be a plasma membrane, which surrounds the cells and protects the cells and helps to make a compartment so that the cells are isolated from other cells and from the environment. Like this classroom, it has walls. This can be like the plasma membrane. So it isolates our classroom from other rooms in the building. Also, the cells have a cytoplasm, which is the fluid that is located between the plasma membrane and the nuclear membrane. The cytosol is the fluid from the cytoplasm, and the cytoplasm includes the organelles or little organs. So the cells have many parts that we call organelles or little organs. And these individual parts, they have a function. We will see what are the different functions of these organelles or tiny organs. And in the cytoplasm is where most metabolic activity take place in the cells. Now the nucleus is the part of the cells that contains the DNA and that is in charge of controlling the function of the cells and directing the synthesis of proteins. So basically it's the command center of the cells, the nucleus. Any questions? So here are the main parts of an animal cell. So here we have the plasma membrane, and then this is the cytoplasm, this is the nucleus, and then we have all of these organelles. Like this is the mitochondria, centrioles, peroxisomes, lysosomes. We have cytoskeletal proteins, we have free ribosomes, we have attached ribosomes to the rough endoplasmic reticulum. And then we have another organelle that's called Golgi apparatus. We have another one that's called the smooth endoplasmic reticulum. So all of these are the main parts of the animal cell. So the cytoplasm, again, contains all the material between the plasma membrane and the nucleus. And the cytosol, which is the liquid portion of the cytoplasm, is made mainly out of water, and then we have solutes dissolved in this water. Like we have ions, calcium, we have magnesium, we have phosphate, sodium, potassium. All of these ions are dissolved in this. And it has a particular pH, certain concentration of hydrogen ions. So cytoplasm has everything, cytosol and the organelles, while the cytosol is only the liquid portion that has all the dissolved solutes. So, cytoskeleton. Cytoskeleton, it is made by proteins, and it takes its name from cytos, which means cell, and skeleton, which means a skeleton, which is a structure. It's a network of proteins or filaments that gives the cells its shape. So we have microfilaments, which are thin proteins. We have intermediate filaments that are not as thin as the microfilaments. And then we have microtubules, which are hollow proteins made out of tubulin. And these will give the shape and the structural support to the cells, and as well enables the movement of the contents in the cell, and also of some cells if they are cells that are able to move. So it helps to maintain pathways through which molecules can move. So it helps in the traffic and organization of the organelles. So now let's look at the chemical composition, structure, and function of the cell membrane. Cell membrane is the same as the plasma membrane and is made out of a bilipid layer, phospholipids. So phospholipids, they have a polar head, which is made of phosphate groups, and then they have two fatty acid tails. Fatty acid tails are hydrophobic, while the polar head is hydrophilic. That's the one that can interact with water. And we have two leaflets, one that faces the external environment and one of the cell, and one that faces the cytoplasm. So the outer leaflet and the inner leaflet. So you have two lipid bilayers. Two lipid layers. So that's why it's called bilayer. Bi means two. And then embedded or attached into this bilipid layer, you have proteins. The proteins can be transmembrane or they can be peripheral. And as well, you have carbohydrates in the plasma membrane. And you have other lipids like cholesterol that gives certain rigidity to the plasma membrane so that the plasma membrane won't be flimsy. So this plasma membrane is selectively permeable. So not everything can come in and out of the cells, only certain items. And then it's dynamic. So we have constant rotation of these molecules within this, or regular rotation of these molecules within the plasma membrane. And some substances can freely pass this plasma membrane, like the hydrophilic ones. The ones that are lipid soluble, like gases, oxygen, CO2, can cross really easily. Ethanol, alcohols are easily transferable between the plasma membrane, and others are not. Others have to cross through these channels. And this helps to maintain or to regulate the environment. Like, for instance, right now, there's only anatomy and physiology 1 students. It's selectively permeable for you. We don't have students from other classes here. So that maintains the internal environment. So here is the plasma membrane, a magnification of a little piece. So this is one of the leaflets. This is the outer leaflet. So this is the bilipid layer. So this is one of the leaflet. This is the other leaflet. So these are the fatty acids facing away from water. So these are hydrophobic. And these phosphate heads are hydrophilic. So they can face the external environment. So this is extracellular space, and this is the intracellular space. And then you have these proteins that are integral proteins or transmembrane. And then the ones that attaches to these transmembrane proteins, either on the outside or the inside of the cells, these will be peripheral proteins. These peripheral proteins can attach or anchor these transmembrane proteins into the cytoskeleton. So here is an example of cytoskeletal proteins. And then these are cholesterol molecules, and these are carbohydrates attached to these proteins. So they form a glycoprotein. Glyco for carbohydrate, proteins for proteins. So these are the components of the plasma membrane. Any questions? So, function of the different proteins. Structured proteins give support and shape to the cells, like the microfilaments, the intermediate filaments, which form the cytoskeleton, or the tubulin. And then the receptor proteins that are found in the plasma membrane detect signals from other cells or from the environment. And then the channel proteins are channels, so they have a pore in the center through which molecules can come in and out. So those will be the channels. And we have different channels. So we have channels for calcium, for potassium, for sodium. So they are specific. And sometimes they have specific names, like the ones that allow the passage of glucose. These are glucose transporters. So here are some examples of the functions of these. So here we have a channel. It's a protein channel. So it has the proteins here. And then this is a frontal cut, so you can see that they have a hollow part center through which molecules can come in and out. In this case, this is in a specific. And then we have carriers. So again, this is a frontal cut showing you these channels. And these channels, they have, in this case, an indentation or a little gap into which a substance binds to this carrier and then later on is transported or dislodged into the cells. So some of them are carriers because they can physically interact with the molecule being transported, while these ones, they don't physically interact. So a channel and a carrier. Now, the receptors, they have an external part that fits a molecule. In this case, this will be a ligand. So the ligand fits specifically into this little part of the protein, and that ligand will make this gate from the channel to open so that the molecules can move across. So an example will be the acetylcholine receptor. Acetylcholine, it's a signaling molecule, or it's a neurotransmitter that once the cells receive this acetylcholine, they can open the gate so that the sodium can move across. And now, some proteins that are embedded within the plasma membrane can act as enzymes. So they can bind substrates, and then they can release a product. And enzymes, remember, they are proteins. Others, they give structural support, like it can be transmembrane proteins attaching to extracellular proteins that are found in the extracellular matrix, or they can be attaching into cytoskeletal proteins. And that gives the strength, so it's like the cell is anchored in both places so that it won't move as much. So they give a structural support. And some others, they link to cells, like this is cell number 1, this is cell number 2. And these proteins forms like little windows. These are called gap junctions. Some cells, they have gap junctions so that you can transfer electrical currents, you can transfer ions like calcium, like in the case of the heart cells, muscle cells, the heart muscle cells, they have gap junctions so that each of these cells will have the same levels of calcium and same levels of electrical current or voltage so that they can contract synchronously at the same time. That's synchronous at the same time. Any questions about the proteins and the plasma membrane? Okay, so what is the difference between intracellular and extracellular fluids? So, intracellular fluid has high levels of potassium and low levels of sodium, while the extracellular fluid is the opposite, has high levels of sodium and low levels of potassium. And as well, it has high levels of chloride. Why? Because sodium attracts chloride. So it makes sense that if sodium is more abundant extracellularly, you have more chloride as well, extracellularly. And the chemical composition for the cells changes according to the cell function and homeostasis. We will see how these changes drive the formation of action potentials in excitable cells like muscle cells and nerve cells. But main changes is that you have more potassium in than out, and you have more sodium out than in. Now, what factors drives the movement of molecules across the membrane? Well, one factor would be the size of the molecules. The smaller molecules cross more easily than larger molecules. They have more kinetic energy because they're smaller. And then the charge of the molecules. Non-polar molecules can cross the plasma membrane, which is nonpolar, at least in the hydrophobic portion, the fatty acid. Examples will be CO2 or carbon dioxide and oxygen. And then the charged molecules, because they have a charge, they have to cross through a channel. Non-polar cross directly the plasma membrane. Polar, they have to go through a channel that will have the opposite charge that they do, partially. And then the concentration gradients. So, a gradient in physiology, it's a difference. Difference can be in concentration, it can be in pressure. So it's a gradient. So molecules will move from high to low concentration by passive transport. I was counting, and you're 16, and I'm 1. Let's say this is the plasma membrane. You are extracellular fluid, you are in the extracellular compartment, you are molecules of sodium, you are in the extracellular compartment. I am intracellular fluid, I am potassium. So, the movement, because the gradient will have to be some of you moving from extracellular fluid, sodium, into intracellular fluid. And the movement will stop when you reach equilibrium, when there are equal amounts of sodium in and out. So then, if you were to diffuse, diffusion is movement. If you were to move from extracellular to intracellular, and you will stop moving until you reach equilibrium, and you are 16 sodium ions, how many of you will have to move into the intracellular compartment to my side, so you will stop moving? You are 16 sodium molecules. You will diffuse, you will move from extracellular to intracellular, and you will stop moving once you reach an equilibrium. Until it's the same amount of sodium out than in. Go ahead with your hands. How many will be? Eight, right? Because it will be 8 sodium there, 8 here. So then that's when you stop the diffusion. And now, you have transport proteins or carriers and channels that assist the molecules that cannot cross a native. So, I mean, we are all physically able in here to walk. But then let's say that they were people that couldn't walk. And you only have 1 or 2 wheelchairs that will help for the people who cannot walk to move across the plasma membrane, which will be here. So the wheelchair will be considered a channel, a transporter, right? And then, how long will it take with 2 wheelchairs to move 8 people from outside of the cell to the inside of the cell? It will take long, right? So that's the limit time, the number of transporters. If we had 8 channels, 8 wheelchairs, 8 transporters, it would take less time to move, right? So that's the point, that sometimes if you have less transporters, or if you don't have the transporters at all, if they are saturated, then the diffusion will be slow. Now, energy use. So passive transport requires no energy. If you're physically able to walk, then it won't take you so much energy to cross, to walk from outside to in. And you're young, everyone here is young, but what if you were old? It would take more energy, right? So energy use, passive transport requires no energy. So in this case, it will be your youth being passive transport, and as we age, it will take us more energy to move. In that case, will be active transport. Active transport requires ATP, adenosine triphosphate. And then the membrane composition. Lipid and protein makeup affects permeability. So, let's say that this is the only area, like in there, or well, over there in that gap, and there you can also move. But what if I start putting something like this, like a protein, and over there as well? Can it be easily for you to cross? Not as easily as if you have it open, right? So in that case, that can be the lipid and protein makeup. So depending on what proteins do you have there and lipids is how easily that can open. Because how, move, because how open is the space. And also electrochemical gradients. So it's not only having a concentration, but also having an electrical charge. Right now, when you come and sign, guys, you come and sign in here. But before, last semester, I had people going there and sign. So that was kind of the electrical gradient, that was the attraction. If you wanted to demonstrate that you were here, that was your electrical charge, so it's positive, telling, well, my professor needs to know that I'm here, so you came and signed in here, that was a positive sign, so you were attracted. Now it's neutral, it's there in the plasma membrane, so you, of course, you just go there. But what if I have here something that attracts you, like, oh, well, I will give you chocolate or candy, and then you come. So that's a positive charge. Or if I'm old man and I'm so intimidating, which sometimes happens to some people, I don't know why, but then, yeah, it's crazy. But basically, if I would be like all stone faced or looking mad, I mean, you wouldn't come and talk to me at all. Right? So that's the electrical gradient. So if you are positive, if you're sodium, you will be attracted by negative charges and vice versa. So any questions about the factors that determines the movement across the plasma membrane? So how does the passive transport processes work? So in physiology, items move from more to less, from more concentration to lower concentration. That's diffusion, movement of solutes from high to low concentration. Simple diffusion, basically nonpolar molecules pass through the lipid layer directly, oxygen and CO2. That's simple, doesn't require a transporter. Facilitated diffusion requires a carrier or a channel protein. So those are for solids. Now for water also moves. Water moves from high concentration of water to low concentration of water, or from a low concentration of solutes to high concentration of solids. That is osmosis. And the reason for that is because water is trying to make the solutes to be in equilibrium. in terms of concentration. So if you have here water in a beaker, let's say you have compartment A and compartment B, and then in compartment A you have 3 solutes, in compartment B you have twice, so you have, well, 7, so 3 solutes versus 7. So water will move from this area, compartment A to B, because it's trying to be at the same level of concentration to dilute the solutes. So in this case, which compartment is going to lose water? Which one is going to shrink? Which one will become shorter? A or B? A. So in this case, you will have this, and what is going to happen with compartment B? It's going to get the water that it moved from A to B. And this is how we actually regulate our water concentration in our body and the compartments where it's located. So let's say that we have the extracellular compartment with increased concentration of sodium. So let's say that we have 150 milliequivalents per liter. And then the intracellular compartment has low sodium as always, the normal level of sodium, let's put normal sodium, and then the typical sodium levels in the extracellular compartment, it's 140 milliequivalents per liter. So if this is the case, your intracellular compartment will move water. So H2O will move in this direction until you reach this normal level. So this is normal sodium. So this is the intracellular compartment, and this is the extracellular compartment. Every time that you eat a salty food like pizza, hot dogs, popcorn, any of those processed foods, and even when you eat something that's sugary like ice cream, that increases your osmolarity in blood. That makes your cells to give water to the blood vessels, so that you reduce the osmolarity and then you reach normal levels of sodium. So our cells dehydrate, and then we start feeling thirsty and we drink water. so that the water will be returned back to the extracellular, to the intracellular compartment. So we feel thirsty because our cells shrink when we have excess of sodium or excess of glucose in our bloodstream. And the movement of water is osmosis. So we have aquaporins in the plasma membrane through which these cells transfer water across the compartments. And here is better example than what I had before. So here is the beaker. You have low concentration of glucose, in this case will be 5%, which is still high. And then in here you have 50%. So these green dots represents glucose. And of course the blue represents water. If you have a plastic divider, and if you remove it, this will move it to here, from 50 to 5. Why? Because there is a gradient. What is the percentage? What is the gradient? What is the difference between 50 and 5? I heard it louder. 45. So it will move from 50 to 5 until everything is going to be what? 55. So everything will become 55. So no net movement of molecules between compartments. But this is solutes. In the case of water, will be happening what I explained to you there. While one compartment increases volume, the other one decreases the volume. So what is the difference between simple diffusion and facilitated diffusion? Well, in simple diffusion, you don't require channels, and in facilitated diffusion, you require channels. And then facilitated diffusion happens for larger molecules or polar molecules, molecules that are charged. In the case of simple diffusion, will be molecules that are not charged, and typically these molecules are small, like CO2 or oxygen or ethanol. So illustrations about this type of diffusion. From the extracellular compartment here on top to the intracellular compartment. So here we have from more to less and from more to less in the different directions. From intracellular space to extracellular space, or from extracellular space to intracellular space. And these are non-polar molecules. So see, you don't have any channels, only the phospholipid bilayer. And then in here, we have channels. So these are polar molecules or ionic compounds. They have a charge, and they use these carriers. They physically interact with these carriers. In this case, this carrier brings or endocytoses the molecules, and in this case, this carrier brings out or exocytoses the molecules. This is facilitated diffusion of polar and ionic. And in this case, this will be facilitated diffusion of ions through a channel. So in this case, we have positively charged molecules and positively charged molecules. But these channels are specific not only in charge, but also in size and maybe in shape, three-dimensional shape. Again, there is no physical interaction, but maybe this ion is larger than this, and that's why it fits only through here. And maybe it has a three-dimensional shape that it doesn't make it fit in here. So that's facilitated diffusion. Any questions so far? So then, for active transport, in which you require energy, you have primary and secondary. So primary, you require ATP to make a pump to function. This function drives ions in and out of the cells. Typical example is the ATP sodium-potassium pump. So this pump won't work without ATP, and sodium and potassium move across. Many anesthetics, they stop the functioning of this pump so that you don't have movement across of these ions, and your excitable cells, like pain receptors, they don't work and you don't feel pain. Local anesthetics. In secondary, you use the energy stored in the ion gradients to drive the transport. You don't use ATP. It is just the movement of these ion gradients, or these ions, sorry, from more to less that drive the transport. So, difference between facilitated diffusion, primary active transport, and secondary active transport. Again, this one doesn't require energy, goes down the concentration gradient from more to less. And then in the case of primary active transport, you have the use of ATP, and in this case, you move the molecules against the concentration gradient. So from less to more. And then in secondary active transport, you can have two types. Symport, in which you move two molecules to the same side, or antiport, in which you move molecules to different sides, like to the outside of the cell and to the inside of the cell simultaneously. For primary active transport, the movement against concentration gradient from less to more, I typically give this analogy in which basically it's like if you are in a building in a large city where it's the time when people go to work, like around 8 or 9 a.m. And the building is very big, it has several businesses, and there are several employees that are trying to get on time. And there are very few elevators. And then when the elevator comes in to a certain floor, it's already packed. And then someone kind of coming, running, and want to go into this elevator. The person itself, it has to kind of make itself a space by pushing against the other ones, kind of, because otherwise, if it's too aggressive, of course the person would be kicked out by the rest. But basically you have to push against. So it's from less to more. It's with an effort, you require energy. In facilitated diffusion, it's like when the elevator is empty, there's no one there, there's no energy to get into the elevator. Or maybe it's the subway. You have seen that in the subway where someone is coming late and the door is about to close and the person forcibly gets into the subway by pushing the other passengers. So osmosis, how is different from simple diffusion? Well, in the case of simple diffusion, just as osmosis is the movement of solutes and water from more to less. But in the case of osmosis, it's water through aquaporins, and then in simple diffusion, it's molecules that are not water, like it can be oxygen, CO2, but osmosis is specific for water. So here we have an example of osmosis. So here is the beaker. Here you have solute, in this case will be glucose, and then this represents water. At the beginning, everything is at the same level for water, and then if you see here, you have more molecules of glucose. So here you have more osmolarity than here. Now, this membrane that separates these two compartments is semi-permeable. Only water can cross, but not glucose. So then with time, compartment A will lose water, and then compartment B will gain water. So this one will become shorter, this one become taller, this column, until you reach same concentration of water. So in ratio, these solutes will be at the same ratio with water than in here. So you stop moving these molecules until you reach an equilibrium. And what is osmolarity again? Is the concentration of solutes in a particles in a solution. Typically is measured as moles per liter. And in the case of our body, we use milliosmoles, which is 1,000 of a mole. Why? Because our concentration of solutes is minute, but still is very important. And then tonicity is the effect of a solution on cell volume. So we have isotonic solutions in which the volume doesn't change. It's iso. Iso means same, or iso. And then we have hypertonic solutions in which you have high level of solutes, and that increases depending on the solutes, the osmolarity. And then you have hypotonic, in which you have low concentration of solutes. With hypotonic solutions, you gain volume. With hypertonic solutions, you lose volume. And isotonic doesn't make you to lose volume. You remain at the same level. So when you put cells in a hypertonic solution, the cells will crenate or shrink. When you put cells in an isotonic solution, the cells do not gain nor lose volume, so the volume stays stable, the shape of the cells stays the same. And in hypotonic solutions, the cells gradually start increasing the volume until the cells can burst. So that's a potential effect. So the cells can burst. This is called osmotic lysis. Or they can swell, but they don't burst. So here we have an erythrocyte, a red blood cell with a disc shape, in an isotonic solution, which is all of this. So this can be 0.85 percentage sodium chloride solution, which is called saline solution, isotonic. So water can come in and out, but you don't win nor lose excess of or water, okay? And now if you put this red blood cell in a solution where you have greater than 0.85% of sodium chloride, water will move from the red blood cell into the external environment, and the cells will crenate or shrink. So you can see the water, how it moves from the cytosol into the extracellular space. And then in the case of when you put your red blood cell into a hypotonic solution with very little solute, you have more solutes inside the cell than out. The water will move in this direction from low concentration of solute to high concentration of solutes in effort to dilute these levels of solute so the cell will start swelling and can burst, can undergo hemolysis. So those were mechanisms of transport and the changes that cells can have in different solutions, isotonic, hypertonic, and hypotonic. Do you have any questions? Now, part of the metabolism of cells comes from getting nutrients from the environment. Or maybe get rid of bacteria or particles that are foreign to the cells. So in this case, we have another mechanism of transport, but includes getting large amounts of items. So you can get rid of items. So this is called exocytosis. You can extrude particles, exo for out, cytos for cell. Or you can bring in particles. So in this case, it's endocytosis, endo for inside. So taking up molecules. And you use vesicles. So you engulf them in vesicles, like you wrap them with plasma membrane, for instance. Phagocytosis is when the cells eat up large particles. We have specific cells in the blood that are phagocytes. So monocytes and macrophages are big eaters. Macro for big, phages for eaters. Neutrophils, eosinophils, and basophils are microphages. They are small eaters. What do they eat? Well, they eat bacteria or foreign particles. If it's a solid particle, it would be called phagocytosis. If it's a liquid particle, it would be pinocytosis. And all of these require energy. and involves moving the plasma membrane. I don't know if there's a picture of a macrophage there, but let's say that this is a bacteria. A macrophage, which is a cell without like a defined shape, is large. It will extend pseudopods or false feet. To ingest this bacteria. So it will wrap it around and bring it in. So that's a macrophage. So it remodels the plasma membrane. Oh well, it is. And it has receptors. So this is a plasma membrane of this macrophage. So it has receptors, it binds this bacteria, and then it extends these pseudopods, endocytoses it, put it in a plasma membrane vesicle, and this now is going to be called phagosome. And this phagosome is going to be fused with this lysosome, so forming a phagolysosome. So with these enzymes, destructive enzymes that you have in the lysosome, you can start breaking down the components of this bacteria. So you lyse it. It's like if someone gives you a steak and then you have a knife, you start breaking it into smaller pieces if you want to make a beef stew, for instance. So you digest or break down this bacteria. And then the next step of this phagocyte is not to keep the waste, to get rid of whatever is not needed, so you get rid of this through exocytosis. And then if it's molecules in the extracellular fluid, you can ingest them together with the extracellular fluid. You form what is called an endosome. And then this is a protein-coated pit. Then you get rid of these proteins, recycle them, put them back, and then whatever liquid you had here or molecules that may be bad, you join them with this endosome, with this lysosome, you break it down, and then you extrude whatever is not needed. So this is pinocytosis. And then you can have receptor-mediated endocytosis, in which you have some receptors in the surface binding a ligand, can be a hormone, and then you form a little indentation here like this to form a vesicle, and then you may have an endosome. You bring it together here with this vesicle, and then you will extract this ligand and then recycle back these receptors into the surface, and then whatever is not needed, you get rid of it. So that's pinocytosis and endocytosis. This is an example of exocytosis in an electron micrograph. So it's extruding. I mean, we bring groceries, right? And then we eat whatever we eat. And if you have waste, where it goes? Into a trash can. So it's like taking away the waste with exocytosis. Okay, so we will leave it up to there. Do you have any questions?